Microbial contamination of feeds. Classification of feed flora, environmental conditions of microbial degradation, and consequences of feed spoilage. So for the classification of feed flora, it's based on the origin. We can have ploughland flora, also known as field flora, such as epiphyta, these are surface microbes, and endophyta, these are inside the plant tissue. And then there is storehouse flora. These are microbes introduced during storage. As well as being based on their origin, they can be based on their multiplication. There is non-proliferating flora and proliferating flora. Non-proliferating flora can be passive or active. If it's passive, it's from the air or other surfaces. If it's active, it's an intentional addition, such as probiotics. Proliferating flora causes nutrient decomposition and toxin production, leading to dysbiosis. So for feed decomposition and microbial gradation, we need to look at the environmental conditions for microbial growth. This includes the initial germ count, which if it's in high amount, it accelerates spoilage, and water activity. Feeds have high water activity between 0.2 to 0.99. High amounts of water activity, also known as AW, supports microbial growth. And then different temperatures can support microbial growth. Thermophiles wanted above 40 degrees Celsius, mesophiles between 25 to 35 degrees Celsius, and psychrophiles below 25 degrees Celsius. Oxygen availability is another factor. Aerobes, microaerophiles, and anaerobes thrive under different oxygen levels. Nutrients are another factor. Sugars lead to organic acids, and lipids and proteins lead to free radicals and biogenic amines. So for the consequences of feed spoilage, we can have organoleptic changes, which change the odor, the taste, the color, and texture deterioration, and this leads to feed refusal, as well as a reduced nutritional value. This includes the loss of nutrients, around 10 to 30% of overall nutrients, and up to 50 to 70% for vitamins like vitamin E. Toxin production is another consequence of feed spoilage. We can have primary metabolites, which are generally non-toxic, and secondary metabolites. Secondary metabolites include bacterial toxins, such as proteins or protein-containing substances, such as endotoxins, and mycotoxins, which are produced by microscopic fungi, such as Aspergillus and Fusarium. And then non-toxic fungi. This is spoilage without significant toxin production, in the case of Trichoderma species. And the fourth consequence of feed spoilage is enteric dysbiosis. This is excessive microbial ingestion, which alters gut flora and disrupts digestion. And for preventive measures, you need to have good storage, maintain optimal moisture and temperature, and aerate feeds to avoid hotspots for microbial growth.